Hello and welcome back to AHA TV, your source for science news and highlights. I'm Kara Moore.
And I'm Trey Johnson. Day 2 of Scientific Sessions 2021 was jam-packed with late-breaking science, networking, and education. At the presidential address, Dr. Don Lloyd Jones, president of the American Heart Association, and CEO Nancy Brown took the stage to speak on launching a generation with ideal cardiovascular health.
The way to keep people healthy was to study healthy people. That is, we should flip the risk factor paradigm on its head and start looking more closely at people who never develop risk factors. Think about that. In the absence of risk factors, CVD essentially does not occur. So if more people could maintain optimal risk factor levels into midlife, CVD would be a rare disease. Specifically, we define CVH as the confluence of 7 metrics: blood pressure, cholesterol, blood glucose, body weight, diet, physical activity, and smoking status, or life's simple 7. But this is more than just data. This is my passion. These data from Dr. Nori Allen show that individuals with high CVH in midlife live 4 years longer overall. But it's even better to know that those individuals also live an average of 7 years longer without any cardiovascular comorbidities. That's right, life's simple 7 gives you 7 extra years of healthy life. Primordial prevention leading to ideal CVH could be our next statin or SGLT2 inhibitor, but with a much broader impact.
At last year's Scientific Sessions, we announced our 2024 impact goal focused on health equity. We are committed to ensuring that all people have the opportunity for longer, healthier lives. This is our North Star. We have bolstered this impact goal through bold actions that we call our 10 commitments. They are anchored by equity and inclusion in research funding, advocacy, community investment, workplace health, scientific publications, and more.
In a series of rapid-fire presentations, results from AHA research on COVID-19 had many attendees tuning in and asking questions. Topics included impact of social determinants of health, repurposing drugs for SARS-CoV-2, and biomarker-guided corticosteroids for targeted treatment of COVID-19.
These data suggest that drugs can be repurposed for the treatment of SARS-CoV-2, and they provide an interactome-based platform for selecting the optimally effective agents.
In summary, the described myocardial gene expression changes, if encoded in the protein, would be prothrombotic by virtue of markdown regulation in the ACE-to-ACE ratio and upregulation in tissue factor. The same changes would produce a pro-inflammatory state due to the ACE-to-ACE ratio being downregulated, and upregulation in tissue factor activation part 2 signaling. Finally, it would be damaging to contractile function by virtue of downregulation of ACE2. As in model systems, these changes are likely mediated by the binding and internalization of the spike ACE2 complex, and as shown, can be observed after mRNA vaccines that encode spike protein. The mRNA vaccine hypothesis is being further tested in a separate NIAID-funded study.
In conclusion, an individualized biomarker-guided approach to corticosteroid use in pneumonia is feasible and safe, with potential benefits including lower cumulative exposure to corticosteroids, increased oxygen-free days, increased hospital-free days. Future larger pragmatic trials with enrichment strategies are needed to evaluate the efficacy and effectiveness of this novel approach.
It looks like platelets can take up SARS-CoV-2 as part of their immune function, but in some patients, that uptake may have 2 adverse consequences. One is the overactive general inflammatory response. We did do proteomic experiments on some of these patients as well. And for the sake of time, I don't have time to show, but it does look consistent with an increase in immune and inflammatory response in those who have an active platelet response to SARS-CoV-2. And in certain patients, this leads to an increase in arterial thrombosis.
Now to a cross-specialty main event. Achieving Health Equity, Advancing to Solutions. It examines why health equity is central to the mission of the American Heart Association and discusses solutions for achieving health equity goals.
It's in our DNA now as an organization to be relentless in our quest for our goals. It is in our DNA to set goals and to achieve those goals. And yes, the American Heart Association has set a goal for 2024 to start working towards achieving health equity and removing barriers, reducing the negative inertia that doesn't allow us all to have a sense of equitable health.
To view each other as equals and as human beings. And I think for the doctors in the audience and all caregivers, that it is really important when patients come to us to ascertain from them, say to them, how can I help you? That's the first and foremost thing to do. And then listen deeply, not move into what you feel they should know, but first listen. And through that listening and that silence, magic can happen.
Dr. Robert Lefkowitz took the virtual stage for the Distinguished Scientists Lecture. He led the captivating presentation on G-protein coupled receptors.
Only partially active. Now, when you have a ligand which shifts this equilibrium of conformations away from the open conformation and more towards the partially activated occluded conformations, you get a Berestin biased ligand. And when you have a ligand which is shifting the equilibrium more towards the open conformation, you get a G-biased ligand. This also further suggests that the two transducers, beta-arrestin and G protein, have very different coupling specificities. Beta-arrestin is much more promiscuous and seems to be able to interact with a variety of conformations, especially these partially activated ones, where the G-biased ligands seem to be much more specific in their coupling and able to couple efficiently only to the open conformation. Now, is this the end of the story? Well, of course not. There's never an end to the story. We're currently using a variety of other biophysical methods, such as crystallography, NMR, and DEAR with other receptors, such as the beta receptor, to gain further insights into these conformational rearrangements, not just at the inner surface, but at other levels of the receptor structure.
AHA is grateful to all our exhibitors and sponsors for supporting scientific sessions. Please visit the exhibit hall to support these sponsors in the same way they have supported our mission. Check out our virtual exhibit hall, browse booths, chat with exhibitors, learn about new advances in treatments, and more.
There is such a wide range of basic science throughout the conference. One evolving area is the application of single-cell transcriptomics technology to cardiovascular diseases and the potential to advance our understanding of biology and disease mechanisms.
The topics proposed in the session, single-cell profiles in cardiovascular diseases, will not only introduce the advanced technology of single-cell transcriptomics, but also provide novel insights into understanding new mechanisms and potential therapeutic strategies in cardiovascular diseases.
Using this multi-omics approach, we learned that T cells were an important immune cells in atherosclerotic plaques, and we identify subset of activated and also exhausted, expressing high levels of PD-1 in plaques as opposed to blood.
We are hearing from the doctors who spearheaded the Doctors with Heart program that connects American Heart Association volunteer cardiologists to clinicians in participating community health centers and federally qualified health centers, or FQHCs, at no cost. This panel discussion, Health Tech and Innovation, will focus on how the program was developed, the need for promoting access to health care, the role technology can provide in health equity, and some early insights.
I really feel fortunate to have come on board with this program at its beginning, at least operationally, as I've been able to take the vision, this amazing vision that was established by Dr. Navarre, by Lynette, and Patrick Waite here at the Center for Health Tech, and to nurture that vision and try to bring it to life in a way that meets the goals it's meant to meet, which is primarily improving access to healthcare.
through all of these visits, Anne-Marie came up with this wonderful idea of Doctors with Heart. And so anyway, we were also the first clinic, and I believe with Dr. Osborne, to actually launch the program. And it was delightful, and it's just an amazing next step to actually prolong these patients to a healthier, longer life.
So we dreamt up Doctors with Heart, which is essentially kind of a matchmaking program between cardiologists who are interested in volunteering their time and free or low-income healthcare clinics that needed access to specialists.
That was one of the key things for us, is we wanted to have different geographies and different populations that we were helping and people that we were helping, so that we could learn as much as we could about what was similar and what might be different.
It's wonderful to be able to just do my little cardiology bit and then be able to help and assist patients and their providers in places where otherwise we'd never have the ability to interact.
There were just really a lot of items that held back that care on the patient. We had one patient show up after being told from a hospital visit that she was in congestive heart failure and she had 5 years to live, and which was just devastating. And it was really wonderful to say, hey, let's not give up. We're in this program now called Doctors with Heart. We have access to excellent cardiologists. We'll get you going there.
There was a lot of troubleshooting to figure out how do we get the best therapies on board in a way that the patient can afford and they can continue to take over time.
That's sort of almost the gravy to this issue. It really is just that putting a person next to another person and provider next to another provider, and then just saying, here's the scenario, here's what's going on, here's the problem.
Dr. Osborne one time made a comment about a specific patient and saying, we're going to add years to his life and he's going to feel so much better. And that, I think I told everybody I saw that for about a week, because that's what it's all about, is having that impact on individual people.
Don't forget to download your complimentary abstracts. Just click on the rotating ad located on the homepage. Made possible in part through support by Janssen Pharmaceutical Incorporated. Dr. Christine E. Seidman is the director of the Cardiovascular Genetics Program and a cardiovascular medicine specialist at Brigham and Women's Hospital. Let's hear a small part of her Science Catalyst keynote and Thomas Smith Memorial Lecture presentation.
I'm also particularly delighted that this is a joint of the Smith and Catalyst lecture, because Tom was a catalyst for science at the Brigham and Women's Hospital, and the fruits of his labor continue today. And what I'd like you to appreciate is that sarcomeres form from the periphery of the cell, where they begin at the protocostomeres, and then through a series of connections with paxillin, alpha-actinin, and titin, they gradually grow centripetally. Well, that was rather interesting to us because of the key junction between actinin and titin. You may know that actinin mutations also cause dilated cardiomyopathy, and these are loss of function mutations. We believe that these early insights suggest, from the human transcriptional data I've told you about, that mutations in different genes can evoke some similar and some very unique responses. And being able to discern these is critical, in my opinion, for being able to develop appropriate therapies for the right person at the right time.
Remember, you can watch this in its entirety on demand through December 15th.
Well, it's time to hear from leadership. Health equity is central to the mission of the American Heart Association, and I know that we touched on that. What were your impressions of the day?
What a fantastic second day of sessions we had on Sunday. There are several highlights, including the late breaker clinical sessions, but I want to really point out the Paul Dudley White International Lecture and Session, which I was pleased to moderate with my esteemed colleague, AHA President, Dr. Donald Lloyd-Jones. I also had the pleasure of moderating a riveting Achieving Health Equity session on Sunday with Dr. Clyde Yancey and Dr. Carissa Baker-Smith on Sunday. Our panel discussion had several key messages embedded in them. One was that a bridge from last year, noting that budgets are moral documents, coined by Dr. Ted Love. The need for investment in health equity, institutional transformation, leadership, the fact that leaders need to be judged on their mentorship and sponsorship, the fact that health equity is actually a quality measure, and that health equity research and activities are rigorous and require a multidimensional approach to training as well as sustainability over time. Be sure to join us for the Quality of Care and Outcomes Research, or QCOR sessions at AHA. Live programming will emphasize global cardiovascular outcomes, learnings from our editors at Circulation, Cardiovascular and Quality Outcomes Journal, and QCOR Awards and moderated networking session. Remember, sessions will be available on demand. Thank you for joining us.
Don't forget to download the mobile app and get started collecting your QR codes for the scavenger hunt. The mobile app, sponsored by Novartis, also gives instant access to up-to-the-minute information about educational sessions, faculty, abstracts, schedule, event information, and schedule planning.
And that's a wrap on Day 2, highlights of Scientific Sessions 2021. Thanks for watching AHA TV. I'm Trey Johnson.
And I'm Kara Moore. We'll see you soon.